Hello, everyone. Welcome to today's live broadcast, Unleashing the Power of Biobanks to Drive Translational Science Forward. I'm Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. Today's webcast is presented by LabRoots.com, the leading social media site for science professionals, and sponsored by PerkinElmer. PerkinElmer is a global leader focused on improving the health and safety of people and the environment. They have a dedicated team of 7,700 employees worldwide who are passionate about providing customers with an unmatched experience as they help solve critical issues in human and environmental health. As a leader in solutions in the translational medicine area, PerkinElmer recognizes the important part played by biobanks and provides tools. The isolation and QC of high-quality DNA and RNA are essential for downstream applications such as next-generation sequencing and qPCR, and the resulting data from these techniques are adversely affected by untreated or poor-quality samples. Furthermore, as the complexity and volume of output data increases, the need for adequate informatic tools becomes a critical factor. For more information, please visit www.perkinelmer.com. Biobanks provide important capacity to collect large genomic data sets intended to enable the study of genetic signatures of an identified population. An essential step in genetic research is the isolation of high-quality DNA or RNA. Many downstream biobanking applications are adversely affected by untreated or poor-quality samples, and large amounts of cellular or other contaminating materials and complex mixtures often hinder many of the subsequent reactions and techniques. Thus, a method for the efficient, reliable, and reproducible isolation of nucleic acids from complex biological mixtures is essential. This webinar presents innovative nucleic acid extraction technologies that help ensure high purity, high yield DNA and RNA. Speakers will also offer techniques for remote analysis and comparison of resulting data by multiple research groups in a biobanking workflow. Before we start, there are a few instructions. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answers welcome too. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can, and we'll follow up if we don't have time today. Want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button on the top right or use the Q&A button. We'll make sure we try to resolve any issues. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Now let's get right to today's presenters. These presentations have been prepared by Drs. Conny Mathay, Andrew Brooks, and Jens Hoefkens. We are proud to welcome them. Dr. Mathay's research background is molecular cell biology with a specialization in dermatology. She holds a PhD in biomedical and pharmaceutical sciences from the University of Namur, Belgium, with publications in fundamental dermatology research. Her background includes traineeships at the Karolinska Institute in Stockholm and the Agricultural University of Gembloux and transdisciplinary applied medical research projects from the Région Wallonie. In 2010, Dr. Mathay joined the Integrated Biobank of Luxembourg as biorefinery scientist. In IBDL, she is deeply involved in lab automation, quality control, and validation of analytical laboratory methods. Dr. Mathay will discuss automation of nucleic acid extraction of 2 biobanking workflows. The Janus KeyMagic automated workstation is a robust and quick magnetic beads-based nucleic acid extraction method that permits you to process different sample types and volumes. The DNA extraction workflows were formally validated from human blood and stool, and this presentation will illustrate the advantages of automated sample handling and extraction of DNA from blood and stool samples and their related formal method validation. RUCDR Infant Biologics is the world's largest academic-based biorepository managing precious biological resources for NIH, foundations, and industrial clients. Andrew Brooks is an associate professor at UMDNJ Rutgers and a molecular neuroscientist whose research focuses on deciphering the molecular mechanisms that underlie memory and learning. Dr. Brooks is a well-recognized genomicist and has been involved in the development and implementation of cutting-edge molecular-based technologies for nucleic acid and protein analyses. Dr. Brooks will present effective approaches to high-throughput quality control of extracted nucleic acids using the PerkinElmer LabChip DS microvolume spectral analysis. A variety of techniques were compared, and data will be presented to show how the LabChip DS provides rapid, accurate quantification of nucleic acids and contaminants with ease and simplicity of spectroscopic techniques and the specificity of fluorescent quantification assays. Jens Hoefkens holds a dual major PhD in mathematics and physics from Michigan State University and oversees the non-clinical life science informatics portfolio at PerkinElmer. Jens has over a decade of experience in running successful bioinformatics businesses with special emphasis on drug discovery, translational research, and cross-omics data integration. Prior to joining PerkinElmer, Jens was the head of the Expressionist business unit at GeneData, where he managed all aspects of the business, including sales, marketing, services, and software development. Now at PerkinElmer, his responsibilities include the newly launched Signals for Translational Medicine Data Management Platform, which has the capacity to create linkages between many types of data, including sequence data and patient data, with potential use in the biobanking community. I will now turn it over to Dr. Brooks.
Great, thank you, Judy, and thanks to PerkinElmer for inviting me this morning to tell you a little bit about what we do to help ensure that biobanking is a responsible practice. And the reason I think that that's important is because as many people are using samples for future use in a biobanking setting and not necessarily for looking at specific analytes immediately, and then saving samples for something down the road, that the focus really now has changed in the community to making sure that quality assurance and quality control for samples is more robust and more reproducible. And one way to do that is to implement technologies that are easily employed across large numbers of labs and large and or small and large biobanks alike, so that when we try to address all of the variables that we have very little control over, which I'm going to describe a little bit, it gives us the ability to actually look at the quality of the sample that we're working on, and not just the nucleic acid quality as we're talking about today, but also making sure it's a correct sample. So one of the things that I would like to start with really is just to give you my view of the difference of what we consider a sample that's biologically stored versus biologically banked. So what is the difference between a stored and a bank sample? And there are really three different things that separate them. First is a bank sample is proactively acquired for future testing and analysis. So this is more to the point that I just made, that now the trend, and it is a responsible trend for translational applications, is that we need to collect larger numbers of samples, and the technologies are so rapidly evolving that we don't know the kinds of analysis that we're going to do. And preparing samples to make sure they're of the quality for our analyses later on is critical for success. Second, a bank sample is often sent to multiple different labs. So unlike an investigator who's storing a sample, looks at one analyte, may or may not remember when it went into the freezer or where it is, and send somebody to go and find it, a bank sample, whether it's formal biobanking or more informal biobanking, is often collected with the intent to send it to multiple different recipients to make sure this is responsible. We need to know that people will get the same quality results for a variety of technologies we might be analyzing. And then the last is a bank sample should never be depleted. So I'm going to show you some workflows, some DNA extraction workflows that have been really optimized to minimize the waste of sample. And the way that you do that is the analytical technologies that we're using for quality control and quality assurance have to be very responsible and use the least amount of sample. So if you think about those concepts and now you integrate that with the fact that we all accept that science and technology is driving innovation, that it's critical that we integrate sample collection, the processing of that sample, and the management of that sample on the eye of this molecular central dogma. Because the days of collecting samples to look at one variant are over and then throwing away that sample, especially in a translational setting. When people collect samples now, they want to not only be able to study DNA at the level of the genome, they say, well, what about if I want to collect RNA from this subject? I want to look at metabolite profiling. I want to look at the proteome. So the onus is on us as biobankers or people that are managing sample collections to be able to prepare and quality control all of the analytes in the context of us understanding these evolving technologies and developing sample-centric roadmaps for future samples and use. A good example of that is working with FFPE. We didn't know what molecular biology was when pathologists were preparing samples for FFPE. We have millions and millions of blocks worldwide. Obviously not the best source for molecular analysis, but something that we have to work with, and something that will continue to be generated. We want to make sure that we avoid those kinds of things in the future on large scale by preparing and storing samples in an appropriate manner that do not limit us to the kinds of analyses that we want to be able to do later on. So just real briefly, the RUCDR Infinite Biologics mission, we enable sharing programs not just for nucleic acid, but for cell lines, tissue, clinical data. Currently, we're the Central Federal Repository for 4 NIH institutes, but we do the same type of work for a large number of disease-related research advocacy groups, and we also have a private sector arm called the Bioprocessing Solutions Alliance that offers the same integrated services for a number of biotechnology and pharma partners. And the goal really is to speed discovery of genes for complex diseases by sharing not just well-annotated samples, but also well-annotated data. And then there are just some metrics on the size and scope of what we do. And before I get into where we identify errors in the kind of technology, it's important to point out that when you're working with a biobank, that there is a lot that goes on for repository management operations in order to have an enterprise-level integration, from managing sample collection through to processing, and then on to analysis, whether you're doing it internally or not. And although technology drives sample management operations, all of these different areas are really, really critical and need to be scalable in order for you to meet the daily flux in sample collections. And this slide is really up there for those that are collecting and storing samples in a translational research environment that may not be formally aligned with a biobank or an institutional program, so that you can appreciate some of the things that will be expected of you in the event that you want to share these samples or make them more globally available. So don't take it personally when a Pfizer or a Merck comes along and you've got a precious set of samples and they say, well, sorry, these are not collected or stored or distributed in a regulated or processed in a regulated manner. We can't include them. It doesn't mean that you need to be disqualified. You just need to think about all of the things that go into this process, and consult and talk with academic biobanks, with even private biobanks, so that you can employ not just the technologies I'm going to show today, but the entire process to make this a responsible practice. And the reason that we have to do this is, and I'm sure this probably is not a surprise to many people, that most of the sample collection errors in a biobank and sample quality issues come from things that happen outside the biobank, before the sample even arrives. Sample swaps, putting the wrong subject in the wrong tube. You would think that if a biobank or a program sent a box with one tube and a registration form, that you had one subject sitting in front of you, you would put the blood in that tube from that one subject, put it in the box, call FedEx, and send it back. People work really hard trying to do the right thing to mess up that process. The other thing is following the instructions. And when you're we deal with a couple of thousand collection sites around the world. We train them, we manage them, we provide all the materials. Most of the errors, 98% of the errors, happen in poor sample handling or mix-ups or things of that nature prior to getting to the biobank. And 2% are issues that happen in the biobank, a sample mix-up, a contamination, or what have you. And that 98% is so much harder to control, and that's why we focus on overall sample quality at the end of the day, because we know, quite honestly, there are some things that we and everybody else that's in this business are not going to be able to control. So instead of beating our heads against the wall trying to accomplish what we will never be able to accomplish, we need to start focusing on the samples we're generating and put more comprehensive and more technologically relevant quality control technologies in place. And as you can imagine, there are a number of different potential areas for adverse sample conditions. It can happen, like I said, the mix-up during collection. It can happen during extraction, and samples do change during storage and pulling them out and distributing them. So the quality control platform that we build needs to be able to address those. So let me show you our DNA extraction workflow and compare this to a lot of labs in the U.S. that are doing this on a small or moderate scale. So let's assume that we're standardizing, and this is a correct assumption in the case of RUCDR, against chemogen extraction chemistry. And we've transitioned to using chemogen extraction chemistry because of our concept of using a single pipeline and the same chemistry, a similar chemistry, and integrating and making our automation platforms more efficient. So the nice thing about working with PE's chemogen chemistry is that we, on any given day, extract samples from blood spot cards, from large volumes of whole blood, FFPE, saliva, bacteria, you name it. And the platform is nucleic acid agnostic, so we use it for RNA as well as DNA. Literally, on our Chemagic Star, it would take from a chemistry competitor literally 4 different instruments to cover the range of volumes and samples that we work with on a given day, where now we can standardize it on one platform. So most labs use kits where they do things manually. If you look at the kind of the old above the dotted line, they don't use vessels that are automation friendly necessarily because they're not dealing with larger numbers of samples. They use technologies that waste large volumes of sample to make assessments. There's a lot of eyeballing that's going on. So I mean that, the way that you find out the volume in your tube is to hold that tube up to the light and look at the gradation, and you can make a measurement plus or minus a couple hundred microliters, right? Not terribly accurate. And then they look at some kind of quality where they may run a gel and say, okay, if we see one high molecular weight band, that's good, the DNA is intact, it's not degraded, and if we cut with a restriction endonuclease, we see a nice banding pattern, we know that that DNA is receptive to enzymatic digestion, so clearly we could PCR, do sequencing, or do what have you. And then that's not incorrect, but how do you know it's the right sample, right? It's a high quality sample, but it's the right sample. So if you contrast that with the workflow below the line, this is an integrated workflow that we run, where all of our extractions are fully automated. Again, we run the Chemagic Star instruments, not unlike the Janus Star, but it integrates the chemistry, the chemogen hardware for bead-based extraction. I think the next speaker is going to talk more specifically about the chemogen platform, so I will leave that to them. We have a platform that we use with Micronic that's been designed and built for biobanking applications. We have 2.8 billion barcodes in our series. These are things that we need to distribute to all of our labs as well. Automation friendly on a variety of platforms. What I'm going to talk about today in more detail is the LabChip DS, which is a technology from Trinian in Ghent, Belgium. That gives us not just accurate quantitation and more sensitive quantitation of nucleic acid, but also does contaminant determination, which is huge for us, given, again, the variety of samples that we start with. We do non-contact volumetric detection using the volume check from Biomicrolab. So in less than a minute, we can read up to 96 tubes that are accurate to plus or minus 3 microliters in a tube, frozen or liquid. This is a huge advantage because even when we're doing automation, we can calculate what the robot's supposed to take, but it's rarely exactly what the robot takes. And it's not because of the precision of the liquid handler, but that droplet on the outside of the tip, evaporation from opening or closing the tube. This is critical for us to know what we have. And then the last thing I'm going to speak about is, that's a picture of the Fluidigm Biomark, where we have developed a technology that allows us to functionally quality control every sample. And I'm going to give you the details on that in a moment. So, so when we think about nucleic acid quality control, we break it down into 2 components. The static biostorage kind of quality control, which we call analytical QC. You're getting your concentration, your purity, your yield, your volume, and functional QC, which is more of a dynamic discovery. And this is where we see the field evolving and the technologies that we've developed in this area. We want to be able to measure sample contamination, whether it was at the time of collection or extraction, predict sample performance. So we know before we generate a sequencing library or do any kind of target enrichment that that sample is going to perform, not just for wasting money on building libraries and sequencing, but for also before we start detailed analysis, okay? And then you can also, if you're smart about it, incorporate subject screening into it, and I'll very briefly show why. So some of the advances in analytical QC are using the LabChip DS. Again, this is a technology that was created and manufactured by Trinian. You can load these cartridges. The LabChip DS can either load in between 1 and 96 samples. You can use individual chips or these plates. You can use multi-channel pipetters. We have it set up so it's automated on a robot. When we load a plate, we can let it sit for up to, an hour before it's run. And I'm going to show you real quickly how the technology works. It does a UV vis measurement. It is a spectroscopic analysis. But the real differentiating factor is the C-Drop software that actually does that contaminant and specific content quantification, which is really critical. So You compare this to competing technologies like NanoDrop. You don't have to do 1 or 8 samples at a time. You can do 96 at a time in about 4 minutes. Our techs take about a half an hour to run a plate with a single channel for a NanoDrop, so it's a huge savings on time. We have robots stamp out these plates and can do thousands of readings. We've done millions of readings on this instrument and let it work at its own pace. And then the data is exportable also with the contaminant data right into our LIMS system. So the LabChip DS requires 2 microliters, although we can get away with a little less, but that's a low volume. You can run it in 16 or 96 well formats. There's on-chip sample preservation, which means you don't have to run it right away. It's loaded. The surface tension pulls the liquid down below the surface into these microfluidic channel. And the other thing is that you don't have any rereads. The efficiency is very high here. So often with something like the NanoDrop, if you don't get a good water bridge or a liquid bridge, you have to go back and reread and then cherry pick. It's a very efficient technology. The order of magnitude for range is outstanding. And there are different flavors of chips that you can use, either single or dual microcuvettes. We use dual for RNA measurements, single for DNA measurements. And the way it works basically is you drop 2 microliters into the port. Again, you do this with either a pipette man, a multi-channel, or a liquid handler. The Janus or any of the other PerkinElmer and Caliper liquid handlers work just fine. The capillary draws the sample in and protects evaporation, so you can let that plate sit, like I said, which is a huge operational consideration. Then you read the empty microcuvettes, and you move the sample onto and read, and it repeats automatically on the second cuvette, right? So this is all internal of what's going on. All you're doing is loading the sample, and the instrument does everything else, and the entire read process takes under 5 minutes. And it's basically a 3-step quantification that we use for doing this. First, you get your traditional A260 reading, so I'm going to show you some data that compares this to other technologies. Then you do your spectral profiling for nucleic acid in the second step. So this is nucleic acids versus potential impurities. Looking at low concentration samples or very concentrated samples, you can distinguish them. And then what you do is you compare your spectral profiling to DNA and RNA derivatives. And I'm going to show you a slide with some data that walks you through this more specifically. This is all automated, and you get the output, and you can customize the output for what you want. So what the C-drop analysis does is it basically deconvolutes the absorbance profiles. So on the left, you can see a reference list. So this is a very simple organic extraction. You want to measure the green, the double-stranded DNA, the potential contaminants are RNA, protein, or phenol. And then on the right-hand side, the actual sample. And this is all automated. You could pull this up on any sample to look at it. But what it does is it gives you protocol-specific contaminant profiles. So we have profiles just for the chemogen kits, for QIAGEN, for Millipore, depending on the ones that you want to run. And PerkinElmer manages these profiles with Trinian, and you can put them on your instrument. And here you can see, here's an example of the green is the spectrum that we're looking to measure, because that's actually a double-stranded DNA. And this is a sample that's got a lot of phenol and a lot of RNA contamination, probably not a good RNase process, organic left over. And what happens is, in this case, you would have had a double, basically overestimation of the amount of DNA that you had that would wreak havoc in downstream applications. You have a very large dynamic range, and the data analysis is repeated. So here's data, and the PDF of this deck will be made available so you can look at it later. Looks at an experiment that we did that looked at potential contaminants in saliva DNA extraction. And saliva is not the cleanest thing to look at. And let's just focus on RNA contamination there in that second column. The blue bar and the red bar are the NanoDrop ND8000, the 8-channel, and the Trinian drop since 96, or the LabChip DS rather. And what you get is the absorbance reading in blue and red, which gives you, just about 325 nanograms per microliter. But if you ran Pico green on that sample, you'd see the actual double-stranded concentration for the dye that intercalates just double-stranded DNA is only 200 nanograms per microliter. So an overestimation of the amount of DNA, which could be very bad. Now, if you run the LabChip DS with the C-drop software, you can see that the purple line in every case for every contaminant lines up almost exactly with the green bar. So what you get is Pico green accuracy and quantity or quantification without actually having to run triplicate samples, doing dilutions, making serial curves, and doing a lot more automation and a lot more cost and time. So this is great for moderate to high throughput labs because, again, in less than 5 minutes, I get very specific DNA concentrations and quality assessments, and I know what the contaminant is in the case of an overestimation, so I can make assumptions and qualify samples for distribution and use. And you can see that's the case in any one of these potential contaminants. It really is. It's an operational changing event within a lab that runs these kinds of samples. The other advantage is working with difficult samples. So we had mentioned FFPE. DNA QC for FFPE can be very tricky, whether you're going to run next-gen sequencing, microarray, or qPCR. Well, this applies here too. We have big questions about data quality, fixation, and storage. So, just because a block is 40 years old doesn't mean it's bad. We have blocks that were created yesterday that are worse than blocks that are 40 years old because nobody changes their formalin, right? I mean, it's a very hard thing to predict. So we have to look at fixation and storage and isolation method as a function of data quality. It's very, very variable. So FFPE QC really requires accurate quantitation and content analysis, or else you are certainly going to have issues with your downstream analysis. So if we look at C-drop versus pico green here, this is a little bit of a different story because it's not always going to line up. So this data, and again, you have more time to look at it later, looks at C-drop versus pico green versus qubit versus a total DNA assay. And this has just happened to be FFPE DNA that was isolated during using a Roche kit. So, no match to pico green specificity. Well, here's an instance of looking at intact double-stranded DNA specific sample. So it's a good match to a total DNA assay, genomic DNA, fragment double strand plus a single strand is included. Bottom line is you may have much more DNA in your sample than just straight fluorescence tells you. And that is critical for working with FFPE because we're starting with such small amounts. So the utility for this technology, being able to fine-tune and address your analysis is really worth its weight in gold for us. So on the analytical side, this is huge. But now we need to look at functional QC. So what are the important characteristics that we're looking at for DNA functional QC? Well, we want to predict sample performance. So we want to know if the sample is degraded, again, if the extraction contaminants impede performance, and predict what's going to happen for downstream analysis, PCR, microarray, next-gen sequencing, so we don't use samples we know are going to fail. We need to confirm sample uniqueness, so the panel that we built repeats in 2 times 10 to the 24th individual, so it truly is unique. And this is very applicable in family-based studies for genetics. We want to be able to identify sample contamination, especially if we're making cell lines from our samples, which take a couple of months versus extracting whole blood DNA. Something can happen during that time for contamination, checking your cell line quality and reproducibility through passages. We want to confirm sample identity, so we can prospectively correct collection errors. All we do now, a lot of labs look at male versus female. Well, that only catches you 50% of the time. We can correct clinical data errors, and then it has to be, something that's efficient. So we developed a technology called SNP-Trace. This is commercially available through Fluidigm. It is becoming a standard for functional QC standardization. There are 96 SNP-type assays. They're all highly polymorphic. They have a set of critical performance SNPs. So if those don't perform, you could know a sample's not going to work well on microarray or on NGS. Don't even waste your time. A lot of them are covered on standard microarrays. So even if you do the best job, there's no collection error, the biobank's great, you send it off to a service lab to do SNP genotyping and they invert plates or contaminate samples, before you analyze that data, you have this panel to compare every sample to make sure that the data you're getting back is high quality before you even go on and start your analysis. It gives you gender, ethnicity, and parentage, so you get a lot of information from this panel. It's very flexible. You can swap out some SNPs and actually use them to screen large sample collections while you're doing your QC, and it's inexpensive. In volume, it could be a couple bucks a sample. And you look at the cost of sample collection, processing, and everything else to know that it's high quality and the correct sample and collect clinical errors and recollect within a time that you can do it is very valuable. You need to have an integrated real-time database that does all the profile comparisons, the sample comparisons, makes all the gender and ethnicity calls. It will give you a pass-fail for sample. The output can be integrated with any LIMS system that you use. We've analyzed well over 1 million samples, and I can tell you our sporadic failure rate in general for genomic-based analysis used to hover around 5%. Some of that is technology-based for analysis, sample quality related. Today, our sporadic failure rate for sample quality after running this and the LabChip DS is about 0.1%. That relates to a huge savings in time and cost for analytic technology cost. So if you look at a snapshot of data, this is just a random 366,000 extractions pulled from our UCDR over a short period of time. and this is with the most stringent analysis cutoffs. And as you can see, for whole blood and lymphocyte cell lines, a very high success rate. But if you look at saliva, buccals, and FFPE, and increasingly more difficult quality samples, you can see that the percent pass rate highlighted in red precipitously drops off, and FFPE is at 55%. Now, this is the most stringent cutoff. So you would expect that for FFPE potentially, but what this allows you to do is fine-tune your analysis, predict your performance, so that you don't have to disqualify sample. There's never been an investigator that says, oh, that's okay, just get rid of that sample, it's no good, right? So you have to work within those parameters. And this could be a very useful tool for setting your own metrics for success. So just in conclusion, the need for biobanking has been defined a long time ago. And with translational applications and personalized and precision medicine, it's only growing at a rapid rate. But quality control must be defined. Functional QC is essential to run a responsible biobank or even collect your samples. Performance prediction is crucial, not just for the cost of the technology, but the analytical cost and the time it takes to only go back and say, oh, well, these samples were no good. And sample identification is vital, right? So it's not just about a high-quality sample, making sure that you have the right sample. And then, the only way to accomplish this really is with integrated sample quality control. And that is by using technologies here, provided by PerkinElmer, in this case for functional QC by Fluidigm, that integrate well into comprehensive solutions, automation platforms that you can use within PE and a variety of different things in order to get to where you need to be. So, there's certainly a lot more data to be shared. Please feel free to contact me, come visit our website. We work with academic and private sector industries alike, so if there's anything that we can do, we're very collaborative. We are a business that's run out of an academic organization more formally, so we're always looking at new technologies and interested in hear people's thoughts, and if there's anything that we can do to help move the field forward and standardize some of this so we can all share samples more efficiently and more responsibly, we would love to talk to you. So I will stop there and pass it on to the next speaker. I think we're holding questions till the end. So.
Yes.
Thank you.
Thank you, Dr. Brooks, for your presentation. Unfortunately, at the last minute, Dr. Conny Mathay got a virus and lost her voice. In her place, we will hear from her lab director, Dr. Faye Betsu. We will also be joined by Gael Hamou. I will now turn it over to Dr. Betsu.
Hello, everyone. So good afternoon from Luxembourg. So as you just heard, the Conny Mathay who prepared this presentation got a nasty virus who destroyed her vocal cords temporarily. So I will be delivering this presentation, and Gael Hamou, our automation specialist, is here with me in case there are very specific technical questions to ask at the end. The objective today is to give you a glimpse on some of our biobank automated workflows for DNA extractions. Before starting, I would like to remind the fact that in biobanks we have, because we're going to talk about biobanking workflows, and we have two types of workflows or two types of methods. On the one hand, we have sample processing methods, and here in IBBL, every sample processing method undergoes a formal method validation. And we have testing methods, as also Andy explained, quality control methods, which are assays, analytical methods. And again, every analytical method or assay undergoes a formal method validation. So the content of this presentation is to show you 3 examples of automated workflows for DNA extraction, which all use the Janus platform. The examples will be extraction from blood buffy coat, the other using a 24-rod head. Second example, again, DNA from buffy coat extraction using a 96-rod head, and the third example is DNA extraction from stool samples. I will also show you some elements related to method validation, as I explained at the beginning. This is something important, method validation, and when we deal with sample processing methods, so this is sample processing where the output is a sample, is not a measurement, is not a result. The output of the method is a sample. So in this context, when we talk about method validation, essentially we refer to validation for the fitness for purpose of the output sample, validation of the reproducibility of the method. So if you put the same specimen and you process it repeatedly, you're getting reproducible results, and finally the robustness of a method. So if This has to do essentially with different pre-analytics. If you stress your biospecimen, different pre-analytical variables, again, are you getting output samples which have the same characteristics. A quick word on IBBL, the Integrated Biobank of Luxembourg, where we collect, every biobank, we collect, we process, we store, we analyze biological samples, we collect associated data, annotations, and these are being made available to researchers. Our collections especially focus on lung and colon cancer, Parkinson's disease. This is our specific disease focus, and other, let's say, specialty of IBBL is the association of the microbiome collection. So for the majority of our disease collections, we also collect fecal, and now we will also start collecting saliva samples to associate microbiome and microbiome signatures to these samples. So IBBL is, of course, the Luxembourg-based biobank dedicated to supporting high standards of medical research. When I say high standards of medical research, a quick reminder about the quality standards here in IBBL. So we are certified to ISO 9001 applied to the biobanking processes and workflows. And 2 weeks ago, we had our audit for ISO 17025 accreditation of our quality control assays, which some of them I will mention. From the next slide, talking about biobanking workflows. Biobanking workflows concern either collection of different samples. This can be either bodily fluid or solid tissue samples. The second bullet point, biobanking workflows can concern production of what we call simple derivatives. Simple derivatives can be aliquots from a primary sample. For example, we centrifuge and we aliquot liquid samples to prepare serum or plasma aliquot. And the third bullet point is biobank workflows corresponding to the production of complex derivatives like DNA and RNA, which is the focus now of the next slides. So for DNA and RNA extraction, for the DNA extraction, we have selected magnetic bead-based technology, so the Perkin-Elmer chemagen technology, whereas for RNA extraction, we work with a silica membrane-based technology. And I will immediately now go to the first example, which is the plasma and Buffy-coat preparation for blood for DNA extraction. When I say plasma, this is EDTA plasma, so plasma for EDTA anticoagulated blood. The process, in summary, is the following. The blood is collected, shipped at IBBL at room temperature. It is processed in less than 2 hours after collection. Centrifugation is at 2000 G room temperature, 10 minutes with intermediate break. And then the plasma gets aliquoted into 12 aliquots of 220 microliters in 0.5 ml cubes. All this is performed automatically on a thickened liquid handler, Evo 200 platform. Now, I mentioned at the beginning the importance of validation. In this case, very quickly for the plasma aliquots, this plasma is validated for fitness for purpose, for downstream proteomic analysis, and any kind of immunological analysis. So, of course, I don't have time today to go into details about this validation process, but essentially we validate plasma relative to stability and robustness as to these downstream applications. Coming to the Buffy-coat preparation, again, this is prepared by the thickened liquid handler, where we receive a fraction of the Buffy-coat, and from this Buffy-coat, then we extract the DNA on NSM1 extractor, Perkin-Elmer chemagen with a 24-rod head. Now, the fitness for purpose of the Buffy-coat is for genetic, epigenetic, or genomic analysis. So, I forgot to click. Yes, this is how the blood looks after centrifugation, where after you retrieve the plasma, you have this Buffy-coat, which gets aliquoted by the Perkin-Elmer. So, again, fitness for purpose of this Buffy-coat or of the DNA extracted from this Buffy-coat, genetic, epigenetic, genomic analysis. This, again, here you can see the picture how it looks. You know that when you collect human blood, you have quite large variations in the Buffy-coat, in the quantity, the viscosity, the texture of the Buffy-coat, and this makes it a challenge to reproducibly pipette the Buffy-coat from from the anticoagulated blood. Now the TCAN platform performs this extraction through a process which you can see here on the right of this slide. So that the tip goes through a spiral movement, performs aspiration of the buffy coat into this spiral movement. When you start from the top, you see this counterclockwise movement aspirating, then going down and performing a clockwise movement, again spiral to aspirate, and we have 6 different levels of this aspiration. All these different parameters during this process are important for the efficiency of the process, the speed of aspiration, the distance between the levels, the 6 levels of aspiration, and so on. Now on the next slide, once the buffy coat. So this is a picture of the TCAN platform, which unfortunately here we had a video, but which does not work. But if you want to watch the video, you can go on the IBBL website and you can see the video there. Essentially what this video shows is the blood tubes. As the process goes on, you have first a barcode scanning of the tubes by the platform, and then you have a level detection, again a laser level detection for the buffy coat, so that the equipment, the automate, knows where, at what level to start the aspiration spiral movement for the buffy coat. And once this is done, we have the buffy coat, which how we validate the whole process, because again I talked a lot about validation. Here you see the data during the initial validation, where you see there had been a first script on the TCAN from 8 ml blood. Now this first script, which was not yet optimized, gave us equivalent results in terms of DNA yield as the manual buffy coat extraction. After optimization of the TCAN process, and when I say optimization, it's all these parameters which I mentioned, the speed of aspiration, the exact movement, the dimensions of the spiral, etc. You can see that we get almost 200 micrograms of DNA instead of roughly 150 from the manual process. So the optimized TCAN script gives us better yield than the manual, and also with, if you see the standardization, also with less variability. The picture is this schematic MSM1 which we use, magnetic bead-based method for extraction of DNA from the buffy coat. These validation results, by the way, have been published in Biopreservation Biobanking, and on this presentation, which I believe will be distributed after the webinar, you have also the references if you are more interested. Again, a picture of the Janus MSM1 equipment. So how the things took place in IBBL. At the beginning, we had the Chemagen MSM1 extractor for the DNA extraction with both 24 and 96 rod head. And only at a later stage, we added the Janus liquid handling platform. So this Janus liquid handling platform was integrated to the Chemagen in 2013. What this Janus liquid handling platform does for us, it does the liquid handling of buffers, magnetic bead solutions, and also DNA samples. It prepares the run for the DNA extraction. It does all the liquid handling again for the DNA eluate to be transferred into the final storage tubes, and also prepares the microplate which we use for DNA quantification. So we don't have a Trinian equipment, as Andy explained. We have more standard spectrophotometer, but the microplate is prepared by the Janus. And so at the end of the process, the output of this process is, on the one hand, a rack containing all the DNA aliquots, and at the same time, the microplate containing the diluted DNA directly ready for quantification. These are more close pictures from the work table of the Janus, where you can see more closely on the upper left, liquid handling, where you put all the buffers. You have the racks which are empty, waiting for the blood samples, so the buffy coat samples to be put in for extraction. You have the sample, the ID 2D barcode scanner, which reads the 2D barcodes which are on the samples. And also there is the spaces I mentioned for the quantification for the plate, the microplate for quantification, and also there is a bottom 2D scanner, which scans the bottom ID of the final tubes with the DNA aliquots. Okay. A very similar, this is going to be very quick, very similar process workflow is applied to extraction of DNA from actually smaller volumes of buffy coat. So the first is which I showed you, is a 24 rod head extraction, which extracts the DNA from 1 milliliter buffy coat. The second here is again DNA extraction, but this time it is from only 100 microliter buffy coat, and this is a 96 batch flow. So it's essentially the same process. The only difference is that it is performed from 100 microliter buffy coat, and it is a batch of 96 instead of a batch of 24. In this case, the average DNA yield is around 14-15 micrograms of DNA from each 100 microliter buffy coat sample. Now I will move to the third example, which is DNA extraction from stool. So again, we use the Chemagic technology for DNA extraction from stool. I told you that microbiome is one of our very important strategic focus here in IBBL. Now, what did we do to validate and to optimize and validate the process of DNA extraction from fecal samples? You can see here we compared 6 different fecal sample collection containers, some of them without any stabilizer. So the brown cup, the yellow cup has no stabilizer, it is plain tubes. Other tubes including stabilizers like the blue and the red, which are stabilizers from Genotech. The second brown from the right side is a Stratec tube, and the first on the right is a tube containing RNA later as stabilizer. So we had these different stabilizers with different volumes of fecal sample included, and we applied 3 different DNA extraction methods to each of the 6 DNA containers. So the MSM1, Chemagic magnetic bead-based extraction method. We applied an all-in-one kit from Norgen, which is a kit allowing a simultaneous extraction of DNA, RNA, protein, and even metabolites. So, and small and big, long RNA molecules. So this is a multi-analyte simultaneous extraction kit. And finally, a kit from Mobile PowerMag, which is again magnetic bead-based method kit, but this time is manual, not automatic. And so we did the DNA extraction. Then the DNA samples, how did we evaluate? We evaluated the DNA extracts in terms of purity, so optical density 260 to 280 and 230. We evaluated them in terms of yield by both spectrophotometry and spectrofluorometry. Indeed, spectrofluorometry gives more accurate results, quantifies double-stranded DNA, where you have always this over-evaluation with the spectrophotometry. And we also measured the exact quantity of bacterial DNA by bacterial qPCR, not AT, qPCR, and the human DNA again by qPCR. We also did an evaluation of potential inhibitors, because as you know, in stool samples you may often have PCR inhibitory substances and contaminants. And so we performed the SPUD assay for the PCR inhibition assessment. And finally, a metagenomic analysis by 16S RNA gene sequencing to see what kind of microbiome profiles we were getting. And now I will show you the results of the 16S RNA gene sequencing. Here you can see the profiles from 3 donors, B1, B2, B3 are 3 different donors, and you can see that each donor has a different microbiome profile, nothing surprising to that point. The next slide is more surprising, because here you see that depending, so these are now samples from a single donor extracted with 3 different extraction kits, DNA extraction kits, and you can see a variability. So this shows us clearly that the DNA extraction method is a critical factor in the 16S RNA microbiome profile that we get from the samples. And you can also see that the 2 methods on the left, MSM1 and PowerMag, which are both magnetic bead-based methods, give us more similar results, which is logical since these are methods which have the same principle, extraction based on the same principle. In terms of richness, so this is now operational taxonomic units, as we call them. In other words, different species or genera of bacteria that were identified by sequencing. So in terms of richness, we have roughly similar richness in terms of numbers and diversity of bacterial species identified. In this table, we have the summary of the different parameters that we evaluated for the validation of our DNA extraction method. And here, you can see that in terms of yield of DNA extracted, the plain tube, so BRW, brown, is this first tube, the brown tube without any stabilizer, extracted with MSM1 technology gives us the higher yield. However, there was quite a big variability in terms of the output of this DNA. And this is something I would like to mention at this point. I think this is something that we observe generally happening in all our workflows. The fact that if you do not use a stabilizer, you have higher variability in your outcome, in your processing method outcome. Whereas if you use a stabilizer, and this stabilizer can be Park gene for the blood, can be a stabilizer for stool, and we have the same phenomenon also for tissue samples if they are collected in specific stabilizers. So maybe, because some of you may argue that by using a stabilizer you get a bias. I'd like to say to that yes, you may get a bias, but this bias is the same, so it's systematic, always the same, and your sample gets stabilized, you get less variability, and so you are the method becomes more robust by using a stabilizer. So coming back here, now in order to see what happens among the tubes with the different stabilizers, this is the one, so the red is, I remind you, a stabilizer from Genotech that was used here. So Genotech stabilizer with MSM1 technology gave us the best results, combining high yield, low variability, good ratio, absence of inhibitors, possibility of home collection, because also that's the other advantage, that people can collect fecal sample at home by using a tube with stabilizer, automation friendly, and finally 16S, because the signature is quite rich. So this was the process that we validated and we applied. Again, so here the picture is the same platform, MSM1, Chemogen, 13 Elmer. For the fecal sample, we have the 24 batch samples in 24 batch. The run takes approximately 1.5 hours for these 24 samples, and the yield from roughly 200 milligrams of fresh or frozen fecal sample, we get roughly 9 to 10 micrograms of DNA. Concentration by fluorometry is roughly 30 nanograms per microliter. So in conclusion, and this is the last slide, for us we find that automation really has significant advantages, and these include the high throughput possibility, especially for our standard workflows. It facilitates the process, and of course it frees time for our lab staff to perform other things. It provides more precise pipetting, and so less variability. This is clear. With automation, we have less variability in the output product. Also, easy electronic tracking. It can be linked to the limbs, and so you have less possibilities for errors in this case. These advantages of automation may include, of course, you can have a possible instrument breakdown. In this case, you must have in place a manual backup solution to process the sample manually. The data integration within the limbs can represent a challenge, and we see this every time. This can be a challenge. In case you don't have really too many samples and you only receive few samples, then of course the efficiency, this is logical, decreases. When you have low throughput, the automation may not be justified from an efficiency point of view, maybe from a quality point of view, but not from an efficiency point of view. And you need staff with software knowledge and some time investment to develop, to test, to validate the scripts. As I showed you in the second, the validation of the script took quite some time and human resources to do that. And a last word from me is that currently we are validating against the MSM1 chemagen technology for extraction of circulating free DNA from serum and plasma. And although these are preliminary results, I can say that when we compare to different other kits and technologies, we are getting the best results in terms of yield of circulating free DNA. And once you have validated all your methods and you apply the one which works best in your hands, the thing is not finished, because we have continuous quality control of the sample processing method. And this continuous quality control involves two things. First, we have internal quality control charts where we continuously monitor the performance of our processing methods. So we have a surveillance to see that, for example, the yield of DNA during this process is consistently the same. And the second way for this continuous quality control is through participation in external quality assessment program at least once a year. So I think that was all I had to say. And this last picture is our laboratory team. And thank you very much for your attention.
Thank you, Dr. Bitsu, for presenting today. Another change, Dr. Hoefkens was called away at the last minute. Dr. Dan Weaver, Senior Product Manager, will present on his behalf. Dan and Jens work closely together. I will now turn it over to Dr. Weaver.
Hello. Today I will be talking about what happens next after the presentations from the prior two speakers. One of the beauties of the modern biobank is they generate tremendous amounts of information. And PerkinElmer has been very engaged in the process of what comes next, what comes downstream of all those data generation activities. So today I will be presenting, first a small safe harbor statement, of course. I'll be presenting about a product we have called PerkinElmer Signals for Translational. So from the PerkinElmer view, there's a perspective on translational medicine that's really driving a lot of our thinking in this space. As was alluded to with the prior two talks, more and more frequently, as patients come in to the lab or come into the clinical setting, they're coming in historically with fairly coarse diagnostic descriptions. And through the application of biobanks and the data that they are generating when they go to some of these high content techniques, we're able to generate a much more thoroughly stratified patient populations. So all these high content techniques, though, the ones that are being used now more and more prolifically throughout the drug discovery, development, translational medicine, and clinical development processes, are generating volumes of data and analytical workflows that are very overwhelming to a lot of the current systems. So when we were standing back and looking at how to help with the data challenges in this space, we began to realize that there is a really underutilized or underserved workflow that we focused in on with our efforts. So today, when a new sample comes into the biobank or a new scientist begins to look at the results from a sample that was collected in the clinical setting, their workflow begins where the instrument ends. And so they begin with taking the raw data, putting it through some kind of a data processing pipeline, and then looking for QA/QC results. And you heard a lot from the prior two speakers about how important proper QA/QC is, both in sample handling, but also in looking at the data that's coming off of the downstream instrumentation and the downstream experimentation. But assuming that those experiments worked successfully, they will then generate some type of secondary analysis where they're looking, for example, for some kind of correspondence or association between various biomarkers and the disease of interest. And then typically, these results are put into some kind of a data silo or some kind of an Excel spreadsheet or PowerPoint presentation that's then consumed, typically used a few times, and then goes into a data wasteland that gets very hard to take value out of. From our perspective, and the driving user for our product efforts, we want to support the translational scientist. And the translational scientist workflow begins in a different way. they don't begin with the results that come off of a particular instrument run. Their workflow begins with a scientific hypothesis. And to be able to support that hypothesis, they want to be able to go in and look for existing subjects and samples in an integrated data environment to be able to answer the question, do we have any left-handed 47-year-old women who have survived breast cancer? Because if we do, I think that there might be a correlation between that and some biologically meaningful result. If they're able to find those subjects of interest or samples of interest, they want to see what processed data exists associated with them and pull those data and the associated secondary results into an analysis environment. And within that analysis environment, to be able to perform integrated analysis, such that the data that's been the data that's been collected, I should say, can be reused with great efficiency going forward over time. And it's this secondary workflow that we see very underutilized, very unsupported within the industry. And this is the driver for what we've created within our product called Signals for Translational Medicine. So this environment is embedded within a best-in-class data analysis tool called Spotfire. And for those who have never used Spotfire, I would strongly encourage you to look for these kinds of off-the-shelf tools for doing data analysis. Within this, our Signals environment is this very intuitive, modern interface for doing data discovery. And then that data, once discovered, can be dropped directly into this analysis tool, in this example, Spotfire. By selecting and doing queries within this, as users perform queries, those queries are updated dynamically within the Spotfire analysis environment. And once they have discovered that set of subjects or samples that are of interest, they can then discover and bring in any related data directly into a Spotfire template. So in this example, the user is discovering some subjects based upon some criteria of interest, and they're able to drop them through a process of selection. They can be, in a very user-intuitive way, point and click to discover or to select the subset of subjects or samples that are of interest by virtue of any of the facets that are associated with those, and bring them down into any type of subsequent statistical analysis that might be able to either refute or support the hypothesis that is driving the analysis. So within this very simple and very intuitive workflow, the user is able to use modern web UI mechanisms for being able to apply a Google-like search. For example, here within the search menu there, they're able to go into the Spotfire interface and using graphic characteristics be able to sub-select the subjects of interest by virtue of any of the data that have been captured and collected. The act of doing this then brings up and applies these to these facets, which then are dynamically refining the set of subjects from the overall set down to the subset that are of interest. And in that process, as they're going through that process, Signals is dynamically giving feedback in terms of these counts as to how many studies are still in view, how many subjects remain, what visits are known for those subjects, and so forth. And so in this way, the user is presented with a vastly more simple and user-intuitive mechanism for being able to discover the data of interest and then drop it directly into Spotfire. To be able to do this, we've had to build a back-end that can easily support a broad set of kind of core big data challenges. And for those who have spent time looking at this, you can actually break this down in a number of fairly specific ways. So within the concept of big data, there tend to be 4 aspects that tend to be dealt with or thought about when you're trying to deal with the large amounts of data that are being generated in this space. The first is, of course, diversity. If you have a broad set of data that's been collected and you want to put it all in one integrated data environment, there needs to be a novel approach to allow it all to fit together. And Signals has come up with a very different way of trying to address the diversity of data that exists within this space and allow it to reside within a single representational schema that's both dynamic and flexible and extensible without having to go and rewrite new database views or anything of that nature. There's the issue of isolation. Typically, in modern and a lot of environments, while the biobank might be integrated, the various instrumentation that was used with those samples is not. And there's a diversity of individualized or separated silos that the data flows into. And our approach, a better approach, is to find a single integrated environment for the data to flow into. There's the issue of complexity. It's all well and good to capture all the data in an integrated environment, but you need to be able to apply mathematical and statistical techniques that are able to deal with the very, very high dimensionality of the space that you're collecting and allow for a regular user to test hypotheses without having to become a statistical expert. And finally, there's the issue of infrastructure. If every center that is going to collect these data build up their entire infrastructure, that is very cost inefficient. More and more organizations are realizing the values, both in terms of ROI as well as things like reliability, scalability, and so forth, of being able to put this infrastructure on cloud environments. And so that's where we have built this towards, is to address those kinds of issues of scalability and total cost of ownership over time. So we were looking at all of these aspects as we went into build a system to help support the industry in this way. The other thing to realize is a lot of organizations focus extensively on the analytics side. And so analytics is very important, of course, but it's only part of the equation. Beneath that, you have all of the other aspects that will be confounding to organizations that are trying to make sense of or derive value out of the data that they've captured. How do you make the data compatible? How do you make it such that it can be readily updated? How do you manage things like access control, particularly if you are a center that is supporting a broad variety of different operations and are generating data for a variety of different projects? Very likely, some of those constituencies will not want their data to in any way be available to others. Where is it located and how is it backed up? As these data volumes grow, the basic issues of data backup and data availability become extraordinarily important. So when you're thinking about the data you're capturing out of your center, be aware of this broad spectrum of issues that are going to come and be confounding as you're successful, as your samples grow, and as data generated from those biobank samples grows. Categorically, these can be broken down, in our view, to three types of problems. Problems of perception, agility, and flexibility. And in the interest of time, I'm not going to dwell on this too much. Suffice it to say that within each of these, you can identify kind of key capabilities or categorical requirements that then correspond to functional requirements that have to be results in whatever ideal system will come out of this process. For example, in the concept of data perception, you need to be able to do data discovery and drop that into places where you can take advantage of the human visual perception as a mechanism for driving more efficient analysis. Context of agility, you need to be able to very agilely integrate data over time, and that needs to be able to include structured as well as unstructured data, and so forth. And finally, in flexibility, one of the key characteristics here is in order for this to be able to be scalable over time, we submit that you really need to be looking at solutions that are able to take advantage of the cloud infrastructure that's becoming progressively more predominant within the organizations and within the industry. And this provides functional outputs like flexibility, scalability, and externalization out of the box. So within PerkinElmer, we of course have items that address each of these characteristics. For us, perception is really driven by the toolset TIBCO Spotfire, which is a best-in-class data visualization and analysis tool. Agility is provided by the product I was mentioning briefly, which is PerkinElmer Signals, as well as a collaboration that we have with an organization called Atibio, which is really a best-in-class unstructured data consuming and analysis tool. And flexibility is provided by a variety of technologies that underlie the technology stack that we use for bringing these data together. SQL-based tools, NoSQL-based tools, as well as Hadoop, are all part of the ecosystem that we had built up to bring these solutions to our customers. So, in a nutshell, PerkinElmer Signals really tries to apply these methodologies and techniques to the problem of the translational medicine space. We would love to talk at greater length about this. I'm just going to highlight a couple of characteristics about the infrastructure and the way we view this problem space. One of the key requirements is the concept of the storage requirements. And if you look at this space carefully from our perspective, you'll see that they can really be broken down into three distinct sets of requirements. One is the management of the annotated raw data. These typically are the large .bam files or .cell files, the large blocks of information that are coming off of the instrumentation. They tend to be relatively few in number. They require processing to extract the value out of them, and the requirements, if you look at these in general, is very similar to that of of a document store. There are also then the annotated process data and the annotated derived data. These are very similar to kind of historically SQL and NoSQL databases, where the data is in very large in cardinality, so there's lots and lots and lots of them, but very small in terms of data volume. What I mean by that is each individual result is small. So you can think of this as the individual the individual variants are found in a VCF analysis of a next-gen sequencing. And from these data, there tend to be lots of branching analysis pathways. Within our vernacular, the way we approach this problem, we tend to refer to these as the measurements. And this concept of measurement crosses all of the various data types that you would see in this space. And finally, you see the concept of entities. These entities are the things that exist in the actual physical world. These are the subjects, the studies, the samples, the visits. These are the physical entities that the measurements are related to. They tend to be relatively few in number, comparatively speaking, but they have a lot of interrelational complexity, so they're related to each other in complex ways. And it is through this breakdown of the way the data is structured that we're able to digest the problem and able to make it tractable to have all these different data types represented in a single environment and single infrastructure. At a very high level, and very briefly, here is a snapshot of the Signals architecture. So it is based in the Amazon Web Service. It is based in, we field this as virtual private cloud to our customers. And from the outside, everything comes to Signals through a series of REST calls, so REST API calls. This affords us a lot of nice encapsulation and nice breakdown between the outside world and the inside world. It allows us to handle very securely things like data access and data control. So everything coming in to Signals is coming in through secured and encrypted REST calls and REST JSON result sets. And inside Signals, then you have a variety of technologies that are brought to bear to address the various aspects of the problem that I was previously describing, using things like Elasticsearch and data ontologies, using a custom-built Signals server for processing the requests and efficiently operating on the data. And within this, we also have capabilities for doing image processing and image analysis using tools like Acapella and Omera. Again, if there's interest, I would love to speak with you more and go into more detail about exactly how we're fielding this and exactly how we see Signals being applied throughout the translational medicine data lifecycle. So in brief, what we have created in Signals for Translational is a very highly flexible physical model that allows a very broad variety of data types to be integrated into a single data store. We've actually at this point applied this to data types, spanning the gamut of translational science, but also including things like basic drug discovery research and development, as well as target validation and analysis. So we believe we have something in hand that allows for just about any type of scientific measurement to be captured. It is based in cloud infrastructure, so it leads to a high degree of scalability and extensibility, and is deeply integrated with a best-in-class data analysis platform, specifically TIBCO SpotFire. And with that, I will conclude, and I think we are ready to shift over to a Q&A section.
Yes, thank you. Excellent presentations. Thanks for bringing that information to us. Before we get started with your questions, here's a quick reminder about how to reach us today. Questions can be sent via the green Q&A button at the lower left of the presentation window. Our first question is, what is the sample performance for punched biopsy tissues? For Dr. Brooks.
What is sample performance? So punch biopsy tissues really are dependent on the fixation. So we use some technologies actually for punching samples or coring samples from FFPE as well as from frozen samples. But if these are fresh punch biopsies, like a TrueCut needle pass or something, the biggest thing is making sure you put the sample in the right preservation agent. So if it's not something that can be processed right away, you can use something like RNA later or some other preservation agent until you can get to process it and store it appropriately. But we find that if the collection is appropriate, the sample performance for punch biopsies is just as good as a surgical specimen coming out of a biopsy. So I hope that answers and addresses the question.
The next one's for you too. For a biobank using acrylamide gel-based methods for fragment size QC, what thoughts do you have on alternatives?
Yeah, so this is a great avenue for lab on a chip kind of technologies. So we use the LabChip GX for these applications. And we do this not just for DNA sizing for QC on PCR product sizes before they go on to other applications, but also for specific sizing of DNA fragments. And really, we're not as interested in looking at high molecular weight for those kinds of applications. It's really for DNA sizing for high throughput following fragmentation, following peak size. These are all critical for all of the intermediate steps for some of the downstream analyses for target enrichment and mostly for next-gen applications. And the nice thing about the GX is, again, we can run few or as many samples as we like without changing that overall cost. It's a rechargeable chip, and we use that as standard QC for sizing for anything that has to do for nucleic acid along the way. And it goes without saying, it's a standard for us for looking at ribosomal subunits as a function of RNA. We run another technology that's qPCR-based to look at the fidelity of transcripts, but certainly as a first pass to see if we're going to have issues or concerned about RNA samples, we will employ the LabChip GX, again, standardizing on one platform for looking at nucleic acid quality.
Thank you. I would once again like to thank Dr. Mathay, Dr. Betsu, Dr. Brooks, Dr. Hoefkens, and Dr. Weaver for the presentations. Do you have any final comments for us today? Okay, I think we will close now. I would also like to thank our as well as our speakers, our sponsor, PerkinElmer, for making it possible to bring this presentation to you. Today's webcast will be available for on-demand viewing through May of 2016. You'll receive an email from us alerting you when it's available on demand and posted on labgroups.com. You're welcome to forward this announcement to any colleagues who weren't able to join in today. Thanks for logging on and participating in today's broadcast. See you next time.